So genetic variation is the differences that we see among individuals. So if you were to go to the grocery store and look at all the people you see, you would notice that everybody's human. We all have the same basic body plans, but we have different variations in color, height, sizes, shapes, and all of that's considered variation. So this is our phenotype, right? The way that we present our inheritance, right? Our genes. But phenotype is dictated by not just the genotype, but also environmental influences, right? Your skin color can be determined by the amount of melanin programmed by your DNA, or how much time you spent in the sun recently, and a combination of both of those. And remember that natural selection can only act on the genetic variation. It doesn't interact on environmental components, right? If you're really tan, that doesn't mean that you're going to have a tan child if your genes aren't coded that way. So the first violation of the Hardy-Weinberg equilibrium is mutations, right? In order to be in Hardy-Weinberg equilibrium, there should be no mutation. Mutation is an accident. So here we can see in this image, we have an original DNA sequence, TAACT, and at some point during DNA replication, this T got switched with a C, and something went wrong. A lot of times we have DNA polymerase that will come in and fix these errors, but sometimes they don't get fixed. Have you ever written a paper and you've done spell check and then you turn it in and your professor still finds typos? Well, sometimes our errors get past our spell check. So now we've got TAACC. And if this change doesn't affect the phenotype at all, this mutation might be neutral and it might not get affected by natural selection at all. If this change is positive, it gives the environment, or it gives the individual some kind of adaptation, then natural selection is going to select for this new mutation. Which is most likely this change is detrimental to the organism, then natural selection is going to select against this mutation. So there's a lot of other ways to get genetic variation beyond just making new genes. Genes that already exist can be duplicated. So in this chromosome, we've got this yellow region here, and it got copied twice for some reason. And now we have twice the genetic material. These can go on to become mutated further. This is actually how we got our ability to smell. So our smell genes have been duplicated many times, and then each duplication allows for more kind of raw material for changes. When you look at mice, they have over 1,200 genes for different odor detecting proteins. Humans appear to have lost this because we only have 380 copies of the gene. And this is likely because humans don't rely on smell. So natural selection has no reason to select for this many odor genes. Mutation rates change in different groups of individuals. So if you look at the graph here, we've got mutation rate on the side, lower at the bottom, higher at the top. And then the size of the genome is just how many nucleotides are in the DNA. Lower at the left, higher at the right. So the higher eukaryotes, plants, animals, fungus are going to be over here, have a large genome, relatively low mutation rate. This is because we have DNA polymerase that will go in and actually correct errors, as well as the fact that we have large genomes and we replicate fairly slowly. Bacteria actually have a pretty low mutation rate, but we see so many mutations in bacteria mostly because they reproduce so quickly. So a generation for a bacteria can be 30 minutes to an hour, whereas in humans, the next generation has to wait a minimum of 13 years to get to sexual maturity, and usually it's much longer than that. Viruses do mutate much quicker. DNA viruses are in red and orange here, and then the fastest viruses are RNA viruses. So things like retroviruses like HIV and the coronavirus, which is an RNA virus. And so these don't have the DNA polymerase working, so they aren't going to have that kind of spell check. And so they tend to have these really high mutation rates with a fairly relatively low genome size. Means that they're changing their genomes rather quickly, allowing for very quick mutation rates. Sexual reproduction is another method of introducing genetic variation. We looked at meiosis in Bio 1, and we have crossing over, right? The sharing of pieces of chromosome from mom and dad. Independent assortment, how these line up on that metaphase plate and determines which way they're going to go. And then random fertilization, which sperm is going to fertilize which egg. And all of these introduce trillions of different options. One thing to note is that sexual reproduction doesn't actually disrupt Hardy-Weinberg equilibrium. The only thing that really disrupts the Hardy-Weinberg equilibrium is mutation, the creating of new alleles. Sexual reproduction tends to maintain the current allele frequencies within a population.